Hello students, welcome to Short Sir classes, and today the question is about a transgene approach and which of the statements that are true for this transgene approach. And here the statements are: first statement that says a tDNA integration that occurs mainly through non-homologous recombination. So this is a non-homologous recombination. Through this process, a tDNA integration occurs. Then in the second option, the gateway cloning depends on recombination technology as opposed to standard use of restriction enzymes and the DNA ligase. Through the gateway of cloning depends of recombination technology. And in the third statement, the localization of beta glucuronidase, that is also called as GAS activity, that is as a result of expression of GAS reporter gene that can be visualized in a histochemical assay, that is in histochemical assay using the XGAL, that is an analog. Now in the fourth statement, that is the green fluorescence protein gene, that is also called GFP, is isolated from the bacterium Photinus pyralis, and according to me this is the correct statement, and where this tDNA integration through non-homologous recombination. And the second one is second option, that is the cloning depends on recombination technology is through the standard use of this restriction enzyme and DNA ligase. So I'm just using my eraser to erase all these things just to get a clear view. Okay, give me a second. Okay, fine, I have a clear scheme now so that I can highlight it again. Okay, fine. So now I'm just changing my pen color. Okay, so now this tDNA thing, that integration occurs through non-homologous recombination, and the second one is the recombination technology of cloning. Recombination technology is standard use of restriction enzyme, that is important, and DNA ligase. These are the two things that are used in this cloning by recombination technology. Now let's see which will be the correct option then. Okay, option A, that is statement 1 and statement 2. Let's see why. Okay, just inserting a page here. So now in tDNA integration, that is tDNA integration, that is through non-homologous recombination, that is through non-homologous recombination, the gateway of cloning system, and that is invented and commercialized by the in vitrogen since the late 1990s. And it is a molecular biology method, that is a molecular biology method that enables the research to efficiency the transfer of the DNA fragments between plasmid using a proprietary set of recombination sequence. sequence. So this is the explanation, and so the correct answer will be this option A. Now moving to the next question. Now this one is really easy. This is about free radicals. So we have to identify the free radicals that marked as question. Here it is. In sequence from the interconversion of reactive oxygen species, that also called as ROS, and as shown below. So this is our sequence. First of all, ROS are chemically reactive molecules. They are chemically reactive molecule. Now moving to the sequence, that is O2, then O2 from what? There is another question. Okay. And then from this H2O2 has formed. Then again a blank. Then from that finally water. So between O2 and H2O2 there must be O2 minus. And then from H2O2 and H2O there must be OH minus, so that it forms water in the final step. So here the options are OH minus, H2O minus, and O2 and here the O2 minus. So here my answer will be O2 minus, that is, and here we have O2 minus, that is our first answer, and the second would be OH minus, that is second statement. So we have, in option A we have our answer, that is statement 1 and statement 2, that is true. So thank you for watching this video.